Hello, my name is Serafina Nieves, and I am an undergraduate senior in biomolecular engineering and bioinformatics. I'm in the lab of Dr. Russell Corbett-Dedig, which studies evolutionary genomics, and today I will talk a little bit about my project on horizontal gene transfer. I study horizontal gene transfer between Wolbachia and arthropods. Wolbachia are bacterial endosymbionts of a broad range of arthropod species. This means that they live within arthropod cells, which includes the cells of organisms such as bees, ants, and spiders. Studies have estimated anywhere from 20 to 66% of arthropod species show evidence of Wolbachia infections. Wolbachia bacterium have interesting effects on the biology of their hosts. For instance, they can change the apparent sex of their arthropod hosts, they can change their reproductive behavior, and they can even change the number of viable offspring that the arthropod has. These properties make Wolbachia a very interesting organism to study, and even an important organism for controlling biological populations of disease vectors, such as mosquitoes carrying malaria. But Wolbachia have other interesting characteristics too. For instance, it has been shown that Wolbachia actually transfers genes from its own genome into the genome of its arthropod hosts. In my project, I sought to identify these horizontal gene transfer events. When do they occur, and how often do they occur? In addition, I looked at what these genes were doing in their new environments. Are they accumulating mutations more rapidly, creating a better, fitter, more efficient protein? Or are these genes being rapidly lost from the genome, almost as if they were never there? I used a variety of computational methods to identify these gene transfers and look at their properties. For instance, I checked the read sequencing depth from whole genome sequencing data. I looked at the gene expression of these transferred genes compared to genes of the host organism, and I also looked at these Wolbachia transferred genes for characteristics of eukaryotes, such as introns. In the end, I found that most genes that transferred from Wolbachia to arthropods were being rapidly lost from the genome after degrading. These genes are accumulating mutations that change the protein at a rapid rate and are doing it so quickly that they basically fall out of the range of our detection. I would like to thank Dr. Russell Corbett-Dedig for his guidance on this project. I would also like to thank the Research Mentoring Internship Program and Dr. Zia Isola for the financial support and the many resources that they provide. I would also like to thank Dr. Angela Brooks for her support of the RMI program.